Very good morning, dear students. In the previous class, we have discussed the various types of enzymes and their classification and what are the general characteristics of enzyme. Now, the most important concept is how the enzyme is acting and how they will speed up the biological reaction. So the next topic mentioned in your syllabus, mechanism of enzyme action, enzyme substrate interaction, the interaction of enzyme and a substrate. So the introductory concept we will discuss in this particular class. So enzyme and substrate interacted by a specific manner, the enzyme increases the rate of the reaction but do not alter their equilibrium constant. That is what we have discussed in the previous class. Enzyme will be increases the speed of the reaction without altering the equilibrium constant. Okay, the enzyme simply speed up the reaction rate at which a reaction proceed towards its final equilibrium. So here you can see there's a simple enzymatic reaction. Enzyme specifically bind with the substrate. The substance in which the enzyme bind is known as substrate. That is very specific. Then once the substrate and enzyme interacted very slowly, once they are in equilibrium, they produce enzyme and substrate complex. Then later they produce the product. A substrate convert into product. Then once the process is over, the enzyme and the product will be detached. This is what the simple enzymatic reaction, where E, S, P represent the enzyme, substrate and the product respectively. And the ES and EP are the complexes of enzyme with substrate and the product respectively. So this is the general and simple enzymatic reaction. And the most important things of this particular mechanism of enzyme action, we should study something called as activation energy. Okay, generally the energy when the enzyme started interacting with the substrate, okay, initially it is, don't think like the enzyme is very specifically immediately it will bind. So it is not like that. The enzyme specifically attach the substrate and check the specificity, and what will happen if and it will check the energy, okay, and there is an energy of activation will be there. In a conventional biological reaction without enzyme, any substance that is interacting with a catalyst, then it requires some energy to activate. Okay, the energy to activate the catalyst, then only it will go and bind, and that energy is known as activation energy. But in the case of enzyme substrate reaction, there is a lowering of activation energy. Say for example, A and B, that is an equilibrium, it will convert it into there is a, when two molecules A and B approach each other, what will happen, equal concentration of A and equal concentration of B should be there to interact. Then they reach an equal stage, that is known as transition state complex. There is a transition between, there is a concentration of this particular product and is almost converted into product. Okay, so that is, then it converted into C and D, and this is known as transition state complex. Okay, so which resembles both the substrate and the product. Okay, and actually when the interaction is happening, something called as activation, the energy required to activate this particular chemical reaction. Okay, the energy is required to bring the reacting molecule together in the correct way to reach the transition state. Okay, so activation energy is the energy that equalize the molecular interaction to reach the transition state. Okay, the transition state complex can then resolve to yield the product C and D in this reaction. Okay, so once the transition complex is produced, then immediately it will produce C and D, and once it is reaching the transition, it's a very slow process, but once it is reaches the transition complex, then this reaction will very fast, it will convert into product. And this difference in the free energy level between the reactants and product is the delta G0. Okay, so there is a difference in the energy. That energy is denoted as free energy level between the reactant and the product. Okay, that is known as delta G0 dash. Okay, and the products are always have the lower energy level than the substrate. Okay, so the product always reach it to the lower energy state than before it is starting with the enzymatic reaction with the substrate. Okay, and what is the mechanism of action of enzyme? Okay, and how this activation energy will be helping. So as I said, A and B will not convert to C and D if they are not supplied with the amount of energy equivalent to the activation energy. So always this A and B will be interacted and converted to C and D if they have an equal amount of energy, the energy equivalent to the activation energy, then only this process will happen. Okay, the enzyme accelerate the reaction by lowering the activation energy. Okay, the enzyme accelerate the reactions by lowering the activation energy. Thus, more substrate molecule will have sufficient energy to come together and form product. This is the main mechanism of enzymatic reaction. Somebody asks you, what is the main mechanism of a enzyme substrate reaction? How enzyme is interacting with the substrate and how they are catalyzing the biological reaction? The one word answer is enzyme lower the activation energy. Thus, more substrate molecule will have sufficient energy to come together and to form product. Okay, so even though the equilibrium constant is unchanged, equilibrium will be reached more rapidly in the presence of an enzyme because of this decrease or reduction in the activation energy. So okay, the equilibrium constant is unchanged. Equilibrium will be attained very rapidly in the presence of an enzyme because lowering of the activation energy will favor this particular reaction. Generally, the enzymes bring the substrate together at a specific binding pocket. The enzyme bind a particular pocket with the substrate is called active site or catalytic site. Okay, then once the enzyme is interacted with the active site, they produce enzyme substrate complex. Okay, the substrate enzyme substrate, they forms enzyme substrate complex. So this enzyme substrate complex will reduce the activation energy. So this is what I have told. If the reaction, how the enzyme is lowering the activation energy, you can see first we should see the energy with the plot without the activation energy. Here the free energy is taking along y-axis and the progress of the reaction is taking along x-axis. You can see this is the plot. Okay, here you can see two reactant A and B, and what will happen, initially this A and B combines together, slowly they'll start reducing, slowly they'll start interacting, and once they start interacting, what will happen, the energy will be getting increased. Okay, slowly it is increased. So once this A and B, it is slowly, once it is increased, and both the energy will become almost equal. Now you can see from here, initially there is a free energy is a little bit high. Okay, for the both the molecule the free energy is a little bit high, and you can see this is once started interacting. So enzyme comes to contact with the substrate. Slowly they are coming into contact and they increase the activation energy. Once they are reached the equilibrium A plus B, that is almost equal, what will happen, once it is there, then immediately it will come to the product. Okay, so this is the energy of the reactant, and finally it will reach into C and D. Here you can see the energy, this difference. Okay, the A plus B, there is a when they reach the initial level, finally the product is reached, and this is known as the delta G, the free energy. Delta G, that is the free energy change, okay, and this is happening without the enzyme. This is a normal chemical reaction. So once two components are reactant and the products are, once it is the reactants are comes to form, slowly they start to increases and that will increase the energy of activation. This is not EA stands for activation energy, energy of activation, okay. So once the sufficient quantity, almost the energy of the activation is balanced, then it is almost, the concentration is almost reached the same, then it will immediately become converted into the product. Here you can see the energy of the reactant will be much low, higher than that of the product. The product has got lower energy than the reactant. So this is the general reaction. Then what will happen in the presence of enzyme? So if you are adding A to B in presence of an enzyme will not allow the molecule to reach this much time. The free energy will not go like this, okay. The enzyme allow the molecule to interact very quickly without crossing this particular step. That means to say, that is what I told, it will reduce the activation energy. Here the peak comes like this, and here you can see the peak only like this actually, okay. Immediately the enzyme and substrate can combine, they can complete the C and D, and they can complete the product. That is a reaction with the enzyme. So this is the major enzymatic reaction, okay, how they lower the activation energy, okay. So that is the thing. Now the mechanism is, we can explain very well by two methods. One is lock and key theory. This is proposed by this is proposed by Emil Fischer, okay. So this is proposed by Emil Fischer. For this one particular things, he got the prestigious Nobel Prize for this particular things, okay. An enzyme can interact with the specific substrate. I told you the enzyme can interact with the specific substrate at a particular binding pocket called a active site, okay. So it may be rigid and shaped to fit the substrate so that the correct substrate bind specifically and positioned properly for the reaction, okay. There is a particular pocket for the substrate, only that particular product they will bind, that position will be happening. Then what will happen, and this particular pocket is known as active site. The specific pocket in which substrate can bind with the enzyme is known as active site. So here you can see the substrate, slowly it will come and bind with the active site, okay. So once it is down the active site, what will happen, it will reduce the activation energy. Then the enzyme substrate complex, immediately what will happen, this will allow the, it will convert the substrate into product actually. So this you can see that once it is done, the product is completely out, okay. So by lowering the activation energy. So this is the mechanism of lock and key mechanism, okay. And why it is known as lock and key? Because the enzyme can be act as a lock and substrate can act as a key, okay. So like that it will be fitting exactly like a key to the substrate, and this is proposed by Dr. Emil Fischer, and he proposed lock and key mechanism. The formation of this particular things is the reason why there is an active site reduction immediately. The enzyme substrate complex is binding with the weak interactions, okay. This is a protein, I told you the weak interaction is happening. Due to the weak interaction they bind and they produce the product. So that is the mechanism of lock and key mechanism. One more theory is known as the enzyme may change its shape when it's bind with the substrate, so that the active site surrounds and fit the substrate induced substrate, okay. So here you can see when the enzyme starts binding, the enzyme will be cooperating with this one, how the shape of the substrate and the enzyme also make a complementary shape to the substrate so that the substrate can specifically bind to the pocket of the enzyme, and that is known as the induced fit model, okay. This is proposed by Dr. Daniel Koshland. That is known as induced fit model, okay. There the enzyme can interact with using weak interaction like a van der Waals forces. I told you these interactions are weak interactions are very, very important, okay. They will produce all those interactions. The formation of an enzyme substrate complex can lower the activation energy in several ways. By bringing the substrate together at an active site, the enzyme is concentrating them and speed up the reaction. So that is what I have told. An enzyme does not simply concentrate the substrate, it bind them so that they are correctly oriented with respect to each other, in order to form the transition complex. Okay, so such orientation lower the amount of energy that the substrate required to reach the transition state. Okay, so such orientation lower the amount of energy that the substrate required to reach the transition state. Okay, so this catalytic site activate and speed up the reaction hundreds of thousand times better than the normal reaction. So this is the way how enzyme lowering the activation energy. An enzyme does not simply concentrate its substrate, it is bind them so that they correctly orient with respect to each other. Then such orientation lowers the amount of energy that substrates required to reach the transition state. Okay, so this catalytic site activate speed up the reaction more than hundreds to thousand times, and they reduce the activation energy. So this is the main mechanism of enzymatic activity. Now, what are the factors which influencing the enzymatic action? There are three important factors which influence the enzymatic action. Number one, the concentration of the substrate or substrate concentration. Next one, temperature. Next one, pH. So these are the main important factors which influences the enzyme substrate interaction. So first one, we will discuss the concentration of the substrate. So this is one of the most important factor, which is the substrate concentration. Substrate concentrations are generally low within the cell. Okay, in the body there will be generally it will be very less. At a very low substrate concentration, an enzyme make a product slowly because it seldom contacts a substrate molecule. Okay, so at a low substrate concentration, an enzyme make a product very slowly because very slowly contacting the substrate. But the substrate concentrations are more, what will happen? The enzyme binds the substrate more rapidly and the reaction velocity is greater than at the lower substrate concentration. Okay, so if the substrate concentration is high, the molecule, the enzyme will be, there will be lot of substrate, there will be competition, substrate will go and bind with the limited active site of the enzymes, and what will happen? There will be much rapid progress than the lower substrate concentration. The rate of enzyme catalyzed reaction increases with the substrate concentration. Generally, the substrate concentration increases, the rate is also increases. But eventually, further increase in the substrate concentration do not result in the greater reaction velocity or greater increase the speed, because available enzyme molecules are binding the substrate and converting into the product as rapidly as possible. Okay, so what will happen if the further increase in the substrate concentration, all the substrate concentration is already bound with the available active site, there are no more active sites are available to substrate to bind, thus the speed of the reaction will be decreases. So whatever available binding sites are present for the substrate to bind, that much and substrate will go and bind, they will convert product. But still, if you keep on at the substrate, that will not be having any change in the reaction, because all the binding pockets of the enzymes are already over, substrates are more. Once the sufficient substrate is binding, enzyme convert that thing into product, there will not be having further increase in the increase in the product or reaction rate. Okay, so what will happen? That particular things we are going to deduce a very important theory called Michaelis-Menten concept. Okay, so this we will discuss in the next class in detail. The enzyme is saturated with the substrate and operating at a maximal velocity. Okay, so when your maximum substrate is added and all the binding pockets are filled by the substrate, then the enzyme is saturated with the substrate. There is a balance between the enzyme and the substrate. No more substrates are required for the enzyme to activate. Okay, and that is the maximum velocity. That maximum velocity is known as Vmax. Okay, so the resulting substrate concentration here you will be getting a hyperbola. We will see that it is useful to know the substrate concentration on an enzymatic reaction need to be function very accurately. So this particular concept, Michaelis-Menten equation, clearly will tell you what is the substrate concentration that is required to operate in an enzymatic reaction in a very ideal way. And there is a constant called Michaelis constant. Okay, that is the substrate concentration required for an enzyme to achieve half maximum velocity. So Km or Michaelis constant, substrate concentration required for an enzyme to achieve half maximal velocity is used as this is the measure of the apparent affinity of an enzyme for its substrate. Okay, it is used an apparent affinity of an enzyme for its substrate. Okay, thus the lower the Km value, okay, the Km value, the Michaelis constant is very less, then the lower the substrate concentration at which the enzyme catalyzes its reaction. Okay, the enzyme with the lower Km value are said to have high affinity for their substrate. Okay, if the enzymes are high Km value, that enzymes have said to be lower affinity for their substrate. This is very important. The concentration required for ideal concentration required for an enzymatic reaction, the ideal concentration of substrate that is required for an enzymatic reaction is first proposed by Lanier, Michaelis and Menten, two Australian biologists actually, and that concept is known as Michaelis-Menten concept or Michaelis-Menten kinetics. So this is what I have told. The x-axis we can take substrate concentration and y-axis what is the rate or velocity. When the substrate concentration is increases, the velocity is also increases. Slowly you can see it is increases. So once all the substrate are binding to the available pockets of the enzyme, then if you add the substrate, it will not go further. It will saturate, and this is the maximum level, and this particular level then it is a constant. Okay, and this is the maximum level. This is known as Vmax. Okay, rate of the product formation when the enzyme is saturated with the substrate. Equal concentration of the substrate and the product that is present, that operate the fast as possible. And in this particular case, and you can see which is the half maximum velocity. If you make a parabola, that is what I told you, this is a parabola or hyperbola. Okay, this is not a hyperbola, this is like this, and you can see the half of this K maximum, that is by K Vmax, that is known as Vmax divided by 2. Okay, that is known as the half maximum. What is the substrate concentration in this particular level? If you plot a line here and if you do a line towards the x-axis, it will end up with one particular point. In that particular point you will get a substrate concentration that is known as Km. This is known as a Michaelis constant. So that's what I told you. This is the constant that clearly tell about the substrate concentration required by the enzyme to operate at half its maximum velocity. Okay, the substrate concentration required for an enzyme to operate half its velocity, because when the substrate is interacted, okay, when the enzyme is interacting with substrate, slowly it will increase. Finally, when the substrate and all the enzymes are binding with all the substrates are binding with the available binding pocket, but still if you add the enzyme, it will not increase further, it will not go like this. Okay, it will saturate means it will be balanced with the binding pockets. How much our binding pockets are available, that much substrates are already came and bound. Then other substrate is also present, they don't have any space to bind, then they will not increase the rates of the reaction, then it will be goes like this. And this is known as the enzyme, the equation called V is equal to Vmax into S divided by Km plus S. This is known as Michaelis-Menten equation, and this substrate concentration study is known as Michaelis-Menten kinetics. So this is known as V is the velocity rate, Vmax is the maximum rate of the product formation when the enzyme is saturated. S is the substrate and Km is the Michaelis constant. That is to the substrate concentration required by the enzyme to operate half maximum velocity, and this is very very important. So this is known as Michaelis-Menten equation. We will see Michaelis-Menten equation in detail about this one. This is all about the concentration of the substrate. Then the next one is the temperature. Okay, the enzyme, all enzymes are protein, as you all know, we have discussed. So obviously it requires an ideal pH and a temperature for the functioning in an optimal level. Okay, that is what we have been telling. The enzyme changes the activity with alternate in the pH and temperature. If the temperature and pH is not optimum or not ideal, they will not function properly, activation energy will not be lower, okay, and it will not produce a adequate enzymatic reaction. Each enzyme function most rapidly at specific or optimum pH, okay, specific or optimum pH, above or below that particular level it will not function that particular level. When the pH deviates greatly from an enzyme's optimum, the activity is lost and the enzymes may be damaged. Okay, when the enzyme, the pH is very very, if the pH is increases, then what will happen, there will be lot of the pH is in the alkaline. What will happen, the enzyme will not go and bind, then there will be a decrease in the enzymatic activity. Similarly, the temperature also. Enzyme have a temperature optima for maximum activity. If the temperature rises too much above the optimum, an enzyme structure will be disturbed and its activity will be lost. Okay, so the enzyme, the temperature rises too much above the optimal level and the enzyme structure will be disturbed. You know that the protein, they are denatured, okay, and it will be losing the activity. If the temperature increases, what will happen? The protein, enzymes are protein, it will be getting denatured. The weak interaction will be broken and the protein, we have already discussed the protein folding. The unfolded form of the enzymatic structure will be, the folded form of the enzymatic structure will convert into unfolded form due to the weak interaction will be disturbed, including hydrogen bond, hydrophobic interaction, van der Waals interaction. All these weak interactions are the stable structures of the enzyme that produce the tertiary structure. Okay, this increase in the enzymatic increase in the temperature causes denaturation, and that may cause the extremes of the activity that will be lower the enzymatic activity. So that is due to either extreme pH or extreme temperature. So the pH and temperature optima of microorganism enzymes often reflect in the pH and temperature of its habitat. Okay, so when you talk about microbiology, enzymes, microorganism has lot of enzymes. Okay, say for example, certain hyperthermophilic bacteria, they survive in that particular temperature range due to the their enzymes are stable. Actually, the enzymes will be stable. Okay, so that particular enzyme can withstand that particular temperature. Okay. So similarly, bacteria growing best at high temperature often have a enzyme with the high temperature optima and greater heat stability. So for example, if you look Bacillus styrothermophilus, Bacillus thuringiensis, all these organisms can Clostridium perfringens. Okay, all these bacteria can have an enzyme. They are having thermostable enzyme, because due to the thermostable enzyme they will be able to function their activity at the high temperature, because their enzymes are also stable at that particular temperature. So this is all about temperature and pH. This is very simple. This is the pH 5, 7, 10. If the pH is taking along x-axis and velocity is taking along y-axis. Okay, so initially when the pH is increases, what will happen? The rate of the reaction is also increases. It requires maximum velocity. Almost you can see nearly it is neutral to some little bit alkaline pH. So once again it is increases, what will happen? The enzymatic activity decreases. Okay, so why? Because the enzyme cannot withstand at the temperature a highly alkaline level. So they will be able to withstand at 7.2 to 7.4. You can see here actually. So this is the velocity. Similarly, the pH. Okay. So similarly, the temperature also, when the temperature is taking along x-axis and the velocity is taking y-axis, this 10, 30, 50, 70, etc. When the temperature is increases, okay, then it will be also increase. The temperature is increases, the velocity is also increases, but when it is reached the saturation level somewhere around 45 to 40 degrees centigrade, you can see approximately here, just below the 50 degrees centigrade, then it reaches the maximum level. But again, if you increase more than 50, then straight away it falls. Okay, then that is the importance of temperature. And pH, the pH is increases, it will reach generally apply neutral to slightly alkaline pH, it will reach. So there will be high velocity, but when you increase the pH further, it will drastically come down. Okay, similarly when the temperature is increases, somehow at some extent it will go and increase. So once it is reach that the particular level, it will decrease. Okay, this is all about the activity of the factors influencing the the enzymatic, the temperature, pH and substrate concentration. So that's all, dear student. Once again we can glance through the main points we have studied. We have studied about the introduction of mechanism of enzyme action. Okay, then we have discussed what do you mean by activation energy, why it is very important, how the enzyme is lowering the activation energy, and the mechanism is a lock and key mechanism proposed by Dr. Emil Fischer and induced with model proposed by Daniel Koshland. Then how the enzyme, always enzyme lowering the activation energy, how the enzyme is lowering the activation energy, and what are the factors which are affecting the enzyme activity. The first one is the concentration of the substrate. There we have seen Michaelis-Menten kinetics. Okay, we have already discussed Michaelis-Menten equation. The next one, the temperature, how it affect, and the pH, how it affect. There should be an optimum temperature, optimum pH and optimum substrate concentration. So this is all about this particular case. So here the entire concept I have taken from Principles of Microbiology by Leninger. Sorry, Principles of Biochemistry by Leninger, you will be able to see. Similarly, Microbiology by Prescott and Pelser also, same concepts are there in a microbiological point of view. And a detailed analysis, you can refer all this textbook. Okay. So thank you very much, dear student. If you are having any clarification, you can write it to me, or you can call me, or you can comment over below. Okay. Thank you very much.